to be. So extra credit when the time comes. Nonetheless, you still need to send me your emails for attendance when the time comes. Let's see. Can someone online confirm you can see the PowerPoint and it looks normal?
Yeah.
Okay. Well, we are recording. We've still got about 30 seconds left. I guess I'll go ahead and start with announcements since we're recording. Yeah, we'll do that. Reminder, independent work is now extra credit. So, I mean, some of you I would really highly recommend doing it anyway because you could really use the extra credit. But yeah, extra credit now. You don't have to do it. So for that reason, too, you won't get that new sheet every week saying, you know, this is the sheet that you do your independent work. You're just going to stick with this last one that came out for week 7. Just write everything there. Do it if you want. Hopefully it'll boost your grade. I guess that's it for independent work. Lab.
But we would still question. We would still have to turn in the 100 though, right?
No, great question. No, that's what I'm saying. No, you do not. That is completely extra credit now. It'll do nothing but it'll help your grade, but it is no longer required. The idea was when I adopted that assignment from one of my colleagues, the idea was that it would help you guys do a little bit extra biology and find stuff that you're interested in. But at this point, so many people are not even doing the bare minimum that I don't even want to worry about putting extra on them. So yeah, it is extra credit now. You don't have to hit the 100. And for that matter, you could go over 100 because, again, it's not required. It'll just help your grade. Every little point you get will help a little bit. So, you know, focus on your labs. That seems to be everybody's biggest problem in here is lab. Not everybody, but the majority. That seems to be the biggest problem. People aren't doing their labs. And I blame myself a little bit. I don't think I was clear with the TA. You're not supposed to leave the lab until you're done with the labs. Like, I don't understand why I'm checking labs and they're not done. Like, you were there in lab. You're not supposed to even leave until you're done with it. So we're going to go back to that. And it's a good time to remind you for labs this week is in person. It's a team effort. Like, we all are collecting data again together like we did on that first lab. So make sure you're there. Make sure you're on time. If you're late, you're going to lose, you'll lose points for being late. If you don't show up at all, you're going to lose a lot of points. If for some reason you missed today, come on Wednesday if you can. What else? Any questions? I guess that's it for announcements. All right, because we are here. This is it. This is the halfway point. I've already or help you make that decision whether or not you want to drop or not. So see me during office hours if you're considering it, because some of you, I hate to say it, but really should consider it. Anyway, let's jump back into it. This is where we left off. We actually started this little bullet point here. We talked about gametes, the life cycle of a sexual organism. But before I want to jump into it, I want to remind you where we are. This whole chapter is basically about cellular reproduction. We talked about the general stuff at the beginning, like the overarching stuff about cellular reproduction. Then we specifically talked about mitosis, and you learned about the phases of mitosis: prophase, metaphase, anaphase, telophase. And I said you should really understand that before you really start studying meiosis, because meiosis is sort of like a different version of mitosis, at least as far as studying is concerned. So make sure you understand mitosis before you get into meiosis. For the exam, a lot of the questions regarding meiosis, because they're so similar, are going to be comparing and contrasting. So as I'm teaching you meiosis, start thinking about how is this different than mitosis, and how is this the same as mitosis. So keep that in the back of your mind. And before we get into it and jump back into what we were talking about, I want to go back to this big bullet point right here, even though we've already talked about it. The homologous chromosome, remember you have 23 pairs of chromosomes in your cells. And when I say pairs, what I mean is you have one of each chromosome, one from your mom, one from your dad. So chromosome number 2, you got one from your mom, one from your dad. Chromosome number 17, one from your mom, one from your dad. So each one of those chromosomes, like number 1 or 17 or 23, whatever it is, those are homologous chromosomes. Homologous chromosomes, again, you get one from each parent, and they carry the same genes. So if there's a gene for eye color on, and I'm just making this up, if there's a gene for eye color on chromosome number 8 that you got from your mom, then on the same chromosome that you got from your dad, on the same spot of the chromosome is the same gene. You might have a different version of it, which we'll talk about later, but you'll find the same gene. And the reason this is so important for the exam anyway is because, again, I keep telling you that you're going to have to do a lot of comparing and contrasting between meiosis and mitosis. Well, that's the first big difference. And I haven't really told you that yet, but I'm telling you now, and I'm going to come back to it too. But the first big difference, or one of the big differences between meiosis and mitosis, remember in mitosis, we split sister chromatids, right? In meiosis, as you're about to learn shortly, we first split homologous chromosomes. So on the exam, whenever there's any discussion about a homologous chromosome for chapter 8, then we're definitely talking about meiosis, because homologous chromosomes are not in the discussion when we talk about mitosis. So keep that in mind. That's your first big hint for the exam anyway. Homologous chromosomes for chapter 8, you should associate that with meiosis. Anyway, that gets us caught up. Let's jump back into where we were. We were talking about gametes and the life cycle of a sexual organism. Already talked about that slide. Here's the slide we left off on. These are some words you need to know. We humans are diploid, meaning our cells carry 2 versions of each chromosome, like I already said. That's what diploid means. And hopefully that prefix, di, hopefully that helps you remember that. Now our sperm cells and our egg cells, aka our gametes, those are called haploid cells because they only have half of the chromosomes. So if you were to open up one of your sperm or egg cells, you would not find 2 of each chromosomes. You would only find 1 of each, because that's part of the purpose of meiosis, as I'm about to get into. It's splitting up those chromosomes because you only want half the information. And I'll come back to that idea here in a second. And that's where we left off. I don't think I got past that. So here we are, caught up. In the life cycle of a human or any sexually reproducing organism, a haploid sperm fuses with a haploid egg in fertilization. So that is what fertilization is. There's a question like that on the study guide. So if we're talking about the life cycle of a human being, we are diploid, but our gametes, our eggs and our sperm, they are haploid. So you get 2 haploid cells, you put them together, and that process is called fertilization. And what is the result of putting 2 haploid cells together, these 2 gametes together? The result is something called a zygote. And that is the first word for attendance today. Zygote. And the zygote is diploid. So we go back to the diploid state. So anyway, there's the words you need to know. You need to know what diploid is. You need to know what haploid is. Hopefully you already know what fertilization is. But you need to know when you have 2 gametes coming together, these 2 haploid gametes, this egg and the sperm, when they come together at fertilization, then the result is something called a zygote, which is then, again, diploid again. Any questions about that slide? The next slide is basically just a picture of all this information. That's a good independent work topic if you want. Like if we weren't in this class and I said, what do you get when you put a baby, excuse me, yeah, some people would say baby correctly. There's a million different ways to describe it. But what do you get when you combine an egg and a sperm? If you weren't in this class, what would you answer? A baby, yeah, because eventually it will become a baby. What about you? What is it? A baby, yeah. See, and I don't know. I've heard all kinds of things. Some people say fetus.
Fertilization.
Fertilization, yeah, that's the process. Some people say, but like the result, some people say fetus, some people say embryo. And actually, all those words have very specific meanings. So if you want to, you can look that up for independent work. Like what comes after the zygote? Is it an embryo? Is it a fetus? Is it a baby? There are actually proper terms. Terms that are really unimportant for the course of this this semester. So that's why we don't get into it. But look it up if you want for independent work. All sexual life cycles involve an alternation of diploid and haploid stages, which is what we just described. Because again, your whole body is diploid, except the gametes that you are producing, those are haploid. And again, when you combine your gametes with someone else's gametes, then you have fertilization, then you get a zygote, and the cycle continues. And this is the important part, not for the exam, but for why we're talking about this. Producing haploid gametes by meiosis keeps the chromosome number from doubling in every generation. So again, we all have 46 chromosomes, right? We have 23 pairs of chromosomes. Each one, we get a mom chromosome and a dad chromosome for each one. If it wasn't for meiosis, then every generation, the number would double, right? Like if I combined one of my regular old cells, my somatic cells, that's a word you need to know, with my wife's somatic cell, then we would have what? 46 pairs of chromosomes. Instead of 46 chromosomes, we would have 46 pairs of chromosomes, right? We would double the information, and that's not good. As a matter of fact, and we'll learn a little bit about this later, having extra chromosomes is usually a really, really bad thing for humans, for animals in general. That's a broad statement. I don't even know if I want to say that. But for humans, generally speaking, extra chromosomes is a very bad thing. For a lot of plants, it's actually a good thing. Another thing you can look up for independent work if you want. But anyway, let's move forward. There we go. So here's a picture of everything we're talking about. There's the life cycle, right? Here's a regular old diploid adults. All those cells, those somatic cells are diploid. Somewhere around here, they're making some gametes. Those gametes are haploid. Then they come together, right? Those gametes, the egg and the sperm, they come together. Again, they're haploid. Fertilization is that process of them coming together, right? That's the verb, right? They're fertilizing. The process is called fertilization. Then we end up with the diploid zygote. That diploid zygote goes through mitosis. Remember, that's how we grow, because you are not one gigantic cell, right? You are a bunch of cells. Even though you started off as one, they kept dividing, and here you are, here we are. That's what mitosis is. And again, you get back to the adult stage, and the circle of life continues. So any pictures, any questions about this picture? Let me put some X's through this, because this is a topic I don't even never talk about that in the exam or the study guide. The N just means like your number of chromosomes, right? So if you're haploid, the number of chromosomes you have is 23. If you're diploid, which is what we are, other than our gametes, we have 2N, meaning we have 2 of each chromosome. So therefore, we have 46 chromosomes. But that is something that will not come up in the study guide or the exam. I don't think. Some of you have done the study guide. Refresh me. That's not on the study guide, right? The 2N thing. And if you don't remember, that's fine. Because I don't remember. Okay. I don't think so either. So yeah, don't worry about the 2N thing. Any questions about this picture? All right. Now we're actually going to talk about meiosis. Here's a picture of meiosis. Before we even get into it, let's mention this. I'll go ahead and say this. Meiosis is 2 rounds, right? Mitosis is just 1 round. It's prophase, metaphase, anaphase, telophase, and you're done. With meiosis, you have to do that twice, which makes sense, right? Because with mitosis, before mitosis, you doubled your chromosome numbers in the S phase of interphase, and then you cut it in half in mitosis, right? So if you double it and then cut it in half, you're right back where you started. But with meiosis, we don't want to be right back where we started. We want to get half of that information. So we double it once, like we did in mitosis, in the S phase of interphase, right? We double it once, like we see here. Then we split that up. So we're kind of back to the original number. But again, we don't want to be the original number. We want to be half. So we split one more time. So that's the overview. Of course, we're going to go into more detail. But that is the overview of meiosis. And again, notice there's meiosis 1 and meiosis 2. And like I said, for the exam, there's going to be a lot of comparing and contrasting meiosis versus mitosis. And not only that, but there's also going to be a little bit of knowing the difference between meiosis 1 and meiosis 2. Now, earlier today, I went back to this word homologous chromosomes. And I told you for the exam, when we're talking about chapter 8, you should associate homologous chromosomes with meiosis only, not mitosis. Because remember, in mitosis, we split these sister. This is basically mitosis. We take the sister chromatids and we split them, right? But in meiosis, we have a round before that, which is where we take those homologous chromosomes and we split those. And then we separate the sister chromatids. So again, homologous chromosomes are the thing that we separate in meiosis 1. That is something you should know for the exam. That is a couple of exam questions right there. In which meiosis, or which stage of meiosis is do we separate the homologous chromosomes? And the answer is meiosis 1, not meiosis 2. Or another difference, another question might say something like, what's the difference between meiosis and mitosis? And one of the options might be in meiosis, you split homologous chromosomes first, and in mitosis, you only split sister chromatids, something like that. Just a lot of different ways you could ask questions. But yeah, the point is, in meiosis 1, we separate homologous chromosomes. We don't get into separating the chromatids, the sister chromatids, until meiosis 2. Anyway, any questions about this basic picture before we jump into the details and fill in these gaps? All right, let's get into it. The process of meiosis. First of all, your book defines it. Meiosis is cell division that produces haploid gametes. And you should be very familiar with this from the pre-lab that we did. There was a few questions about this. So you need to know that meiosis produces haploid, right? So if we're talking about mitosis, and that's really hard for me to do, so I'm really going to abbreviate. If we're talking about mitosis as opposed to meiosis, then we don't have haploid, we have diploid. And I'm going to, again, abbreviate, diploid. Now, if we're talking about meiosis, which we are, then we have haploid instead. It resembles mitosis, except there's two major differences, and you should know these for the exam. The first major difference that your book lists is that the chromosome number is cut in half. And I already said that earlier, 3 seconds ago, right? Not 3 seconds, but a couple slides ago, I mentioned with mitosis, you have your set of chromosomes, right? Your cell is just doing its normal thing in interphase. Then it doubles that amount in the S phase of interphase, and you don't want double the amount, right? So you go through mitosis, and that cuts it back in half, back to the original number. In meiosis, you double that amount, right? Then you go through meiosis 1, so you cut it back in half, or you cut it in half and go back to your original amount, but then you cut it in half again, so you end up with half the number that you started with. So you need to know that. That is one big difference. The chromosome number is cut in half in meiosis, not in mitosis. And the way this is done is because, and I've already mentioned this, the cell undergoes 2 divisions, not 1. And again, this last bullet point, I've already basically said it, just in different ways. Because you had 1 duplication followed by 2 divisions, that means we have half the number. But here's a key number that you need to know, a good way of distinguishing the difference between meiosis and mitosis. 4 daughter cells in meiosis. And again, that was a question on the pre-lab. Which one gives you 2 cells? Mitosis. Which one gives you 4 cells? Meiosis. Which one gives you diploid cells? Mitosis. Which one gives you haploid cells? Meiosis. And I don't know why this isn't on the slide. Maybe it's on the next one. But which one gives you genetically identical cells? Mitosis. Which one gives you genetically unique cells? Meiosis. And then finally, which one is used for asexual reproduction? That's the old one, mitosis. Which one is used for sexual reproduction? That is this one, meiosis. So any questions? All right, let's move forward. Oh yeah, this is the big one. The second big difference between meiosis and mitosis is this, crossing over. Now this is an important concept because this is one that kind of has implications past chapter 8. So when we talk about genetics, when we talk about mutations, and we talk about evolution, this crossing over is an important thing. What is crossing over? It is the exchange of genetic material between homologous chromosomes. It's basically when you swap chunks of chromosomes. So I keep saying that you have 46 chromosomes, right? You have 23 pairs of chromosomes. For each chromosome you have a version from your mom and a version from your dad. That's what this picture is here, right here. Let's just assume the blue is from your mom and the red is from your dad, right? So these are completely separate chromosomes. However, in meiosis, they get really close to each other. This is what this picture shows here. And what will happen is they will literally swap big old chunks of chromosome. So what you end up with is this mom chromosome with a dad chunk, and then this dad chromosome with a mom chunk. And that'll be very important later on when we start talking about genetics. So this is not trivial. Another thing I should point out is this is just one picture of a way it can happen. First of all, crossing over doesn't always happen. It often happens, but it doesn't always happen. So you might end up with a completely mom chromosome and a completely dad chromosome. Or you might end up with this, like I said, a mom chromosome with a dad chunk and a dad chromosome with a mom chunk. Or it could be crossing over twice. Maybe you have that little chunk and maybe one up here. You can cross over twice. And sometimes maybe it's just a little bitty tip at the very end. Or sometimes it's a big old chunk. It's completely random. Like I said, sometimes it happens, sometimes it doesn't. Sometimes it's multiple. Sometimes it's a little bit. Sometimes it's a big bit. For the purposes of Chapter 8, all that information I just gave you isn't important. But that will be important a little bit later as we move forward. So that is what crossing over is. Another important thing you need to know about crossing over, at least for the exam and for understanding Chapter 8, is this bullet point right here. Crossing over occurs during the first prophase of meiosis. You need to know that for the exam. But honestly, if you forget that after the exam, you'll be okay. You do need to know what meiosis is, but the fact that it happens in prophase of meiosis 1, that's a little bit less important in the grand scheme of the semester. So are there any questions about this slide? All right. The next word for attendance, I'm not even going to say it. I'll just circle it. I like to do that every once in a while, as you guys know, so you can read it. So if anybody's laying on the couch with their eyes closed listening to the lecture, I'm sorry to disturb you, but you need to open your eyes and look at the screen. That is the next word for attendance. Anyway, if there's no questions, let's move forward. Let's look at these pictures. Here's interphase. Interphase should look normal. It kind of does, right? Here you have a fully enclosed nucleus. Obviously, that's interphase, right? Here you have all this uncoiled chromosomes, a big squiggly mess. You can't tell one chromosome from the next. Here you have the centrosomes just doing their own thing. They haven't formed the mitotic spindle yet, right? That's just regular old interphase. Let's move forward to prophase. So this should look very familiar. What happens in prophase? The same thing that happens in prophase and mitosis. You got to get rid of the nuclear envelope, right? If you're going to have chromosomes going from one end of the cell to the other, then the thing that holds them, you need to get rid of it. So you get rid of the envelope. If you're going to move chromosomes, then you've got to get the thing that moves them. Remember, the mitotic spindle. So that forms. It attaches, right? So that is prophase. It's the same. And then the chromosomes start moving towards the center of the cell. And then what happens when they make it to the center of the cell? That is what we call metaphase, right? So far, all this is the same. Here's the only difference. Here, when these chromosomes are lined up in the center of the cell, we are talking about the homologous chromosomes, not the sister chromatids, right? The homologous chromosomes are lined up in the center of the cell. It's a tug of war right now, just like it was in mitosis. Eventually, the tug of war breaks them, so to speak, and they actually do get separated. And what happens when they are separated? Then we consider that anaphase. They have been separated. And again, it's the homologous chromosomes that have been separated, not the sister chromatids. The homologous chromosomes have been separated. They're moving towards the other end of the cell. What happens when they make it to the other end of the cell? Just like mitosis, that is what we consider telophase. Once they're at the other end of the cell, that is telophase. And notice too, here we go through cytokinesis. We're turning that one cell into two. So there we go. That's meiosis 1. Any questions about meiosis 1? And notice the big difference. Again, this will be on the exam. What is the big difference between meiosis 1 and mitosis? It's the fact that we're dealing with homologous chromosomes. We're not dealing with sister chromatids. All right, any questions about that? Let's move forward. Look at meiosis 2. Before I even talk about this, let me just say this. Meiosis 2 is essentially the same as mitosis. Because remember, the big difference we were talking about on the last slide was the fact that we were dealing with homologous chromosomes and not sister chromatids. Here, we're dealing with sister chromatids again. Except now we have two cells, right? We start with 2 instead of 1. But the same thing happens, right? The mitotic spindle forms, the nuclear envelopes still disappeared. They start moving towards the center of the cell. What happens when the sister chromatids are in the center of the cell? That's what we can call mitosis. It's a tug of war. Eventually they break. What do we call it when they're finally separated? We call that anaphase. And then at that point, they're moving towards the other end of the cell. What do we call it when they make it to the other end of the cell? That is called telophase. So again, the big difference here, or there is no big difference here. Mitosis is essentially the same. Excuse me, meiosis II is essentially the same as mitosis when it comes to the process. Of course, we're ending with two different things, right? The end of at the end of mitosis 2, we have 2 here, 2 here. So 2 plus 2 is 4, right? We end up with 4 cells instead of 2. So there's a big difference. Trying to think of some test questions from this slide. Yeah, one good test question might be something like, which is more similar to mitosis, meiosis 1 or meiosis 2? And the answer is meiosis 2. That's it, really. So you can see why I kept saying you should study mitosis before you study meiosis, right? Because the way I teach meiosis is like, look, this is just like mitosis, except here's how it's different. So, any questions about this? All right. So I've already told you what's important for the exam. Keep all that stuff in mind when you're studying. What's important for the grand scheme of the semester is this right here. And I've said it before, and I'll keep saying it. What we have here is 4 genetically unique cells. None of these cells are alike. Each one of these cells is different from each other. That is very important. Without that, we will not have evolution. We will not be here in the way we are without that. So that'll be important later. Any questions about this slide? All right, here's some pictures. There's meiosis II in a lily cell. So here we go. This is in your book. I'm teaching out of your book. Now, really quickly, we're going to compare mitosis and meiosis again. Because, again, a lot of your test is going to be about comparing and contrasting the two. For both mitosis and meiosis, we start off with this, with the chromosomes duplicating, right? That happens in the S phase of interphase. We know that. That is the same for both of them. However, the number of cell divisions is what is different. Because, again, if you start off with X amount of chromosomes, and then you double it, and then you cut that in half, right? Then you're just back where you started, which is what mitosis is. But with meiosis, the goal here is to have half the number of chromosomes, right? Because you're trying to produce egg and you're trying to produce sperm. So you don't want the full number, you want the half number. So what do? You double it, just like you did in mitosis. You cut it in half once, just like you did in mitosis. And now, again, you're back to the original number. But this time you cut it in half again. And again, now you're left with half the number of chromosomes. So yes, mitosis, 1 division. Meiosis, 2 divisions. Mitosis, 2 cells. You start with 1, end with 2. Meiosis, you start with 1, you end with 4. Mitosis, you end up with diploid cells. Meiosis, you end up with haploid cells. Mitosis, you end up with genetically identical cells. Meiosis, you end up with genetically unique cells. Mitosis is used for asexual reproduction. Meiosis is used for sexual reproduction. Meiosis, you deal with homologous chromosomes, at least in the first part, right? Mitosis, only sister chromatids. Any questions about this slide? Again, this is just a review. It's just all there on the same side for you. All right. Again, this is still a review. The events that are unique to meiosis occur in meiosis 1. Another way of saying that is the way we are studying this, meiosis 2, not 1, is essentially the same as mitosis. And again, unlike mitosis, meiosis 2 yields haploid daughter cells. Not diploid, but haploid. And if that gets confusing, just try to remember just the basics of sex, right? Common sense should tell you that if you're going to bring a sperm and an egg together, then you're going to want to have half the information, right? You don't want to have a full set of chromosomes in the sperm and the egg, because by now humans would have, I don't even know, I mean, thousands of chromosomes, right? Millions, if every time it doubled, right? So you have to cut it in half each time. So any questions about this slide? All right, moving forward. Same thing, it's just one picture all together. And again, I'm going to put an X to this in thing. And again, we're just looking at the difference between mitosis and meiosis. Notice we're starting with the same thing. Of course, in this explanation, we don't have a picture of the full 23 pairs of chromosomes. That would be insane to try to keep track of that. But we do have two pairs of chromosomes, right? We got the blue pair, the red pair. So there you go. They both duplicate in S phase of interphase. Here, you have the duplicated chromosomes just doing their own thing. Here, remember that was one of the differences. In prophase of meiosis I, those homologous chromosomes are coming together and they're possibly crossing over. There's a good independent work topic if you want to look into that. What even makes that happen? What even brings those homologous chromosomes together? Or, I don't know if you want to look at it a different way, what prevents them from coming together in mitosis? Because they're all there. Like, why is it? What is it about mitosis that keeps the homologous chromosomes from coming together? Anyway, that's deeper than we need to get into for this class, but you can look it up if you want for independent work. So again, in mitosis, we've got these sister chromatids that are lined up in metaphase. Not in meiosis I. In meiosis I, again, it's the homologous chromosomes that are lined up in metaphase. Right? The one from your mom, the one from your dad. And this will actually be an important concept too a little bit later when we talk about genetics and evolution. I'll come back to it. Just know that metaphase I will be important again. Anyway, in mitosis, you separate those sister chromatids. There you go. That's the end result of mitosis. You're back to the original chromosome number. Meiosis, you do it once, you do it again, and now you have half the chromosome number. And again, now look, each of these chromosomes is different now. Each of those chromosomes has gone through crossing over. So it's no longer a chromosome with a mom chunk. Anyway, any questions about? All right. Here we go.
I think somebody online raised their hand, might have a question.
Oh, shoot. I can't read it. Could if you're online, hopefully your microphone works. Can you say your question? Oh, well, I'm sorry.
I'm sorry. I didn't mean to click that. I didn't even realize it was sitting there.
Oh, that's okay. No need to apologize. I'm just glad we didn't miss your question. All right. So like I kept saying, well, I won't go back to that yet. But here we go. This next bullet point right here that we're about to get into, this is an important bullet point regarding the rest of the upcoming information for the semester. As far as Chapter 8 is concerned, we're kind of done with the meat and potatoes. Everything above this is what's important for Chapter 8 for the exam anyway. What we're about to talk about has larger implications for understanding what we're going to talk about when we talk about evolution and genetics. So it's very important stuff we're about to get into. And if you really want to understand like COVID-19, right, the virus or the vaccine, like we don't get nearly deep enough for you to truly understand it, but this is the basic information you need to understand how viruses work and understand how mutations work and how viruses, how you started with Delta and ended up with Omicron and things like that. This is the basic information that you need. So this is very important for your day-to-day life. Here we go. The offspring of sexual reproduction are genetically different from their parents and one another. So we've already said this. We're saying it again. You already know this, right? The offspring are completely unique. Even if you look just like one of your parents, you're not genetically the same as them, right? You are not a clone of either one of your parents. You are a mix of both of your parents. Even if it's not visible, even if you look like one more than the other, you are still a mix of both of your parents. So then the big question is, how does meiosis produce genetic variation? And again, this is a very, very important topic. Without this genetic variation, we don't have all the different weird things you're about to learn about when it comes to genetics, all the different rules and the way things work wouldn't happen without this. And without all that, we wouldn't have evolution. And that's the next portion of the semester. We'll start talking about evolution. And without evolution, we wouldn't be here. So again, this is just a introductory slide. I would ask you if you have any questions, but it's just an introduction. Let's talk about the origins of genetic variation. There's two basic ones that we're going to talk about now. We've already talked about crossing over, so I like how your book we'll come back to that. But let's talk about the new one. Independent assortment of chromosomes. What in the world does that mean? Let's talk about it. When the chromosomes are aligned in metaphase 1, not 2, but 1, and if you remember, if we're talking about metaphase 1, then we're talking about homologous chromosomes, right? So when they are aligned, the side-by-side orientation of each pair is completely by chance. So in your mind, if you can, try to remember that picture I just showed you, where there was only 2 pairs of chromosomes, right? And they were lined up. They had the homologous chromosomes lined up. Looked a little bit like that. I wish I had different colors, but they were lined up. It was metaphase 1, where the homologous chromosomes were lined up next to each other. And if I remember correctly, that doesn't matter. But both sides were lined up. All right, so maybe it was blue on one side and red on the other. But it was both, right? So this one, both of these would be blue, both of these would be red. What we're saying here is that it doesn't always happen that way. It's completely by chance. It's not like when your chromosomes line up in metaphase 1. It's not like all your mom chromosomes line up on one side and all your dad chromosomes line up on the other. It doesn't happen that way. Matter of fact, it happens every which way because there's nothing making it happen. It's completely by chance. So let's talk about some numbers. For a species that has 2 pairs of chromosomes, like we do, every pair orients independent of each other, which is what I just got finished saying. So that means since we have 23 pairs of chromosomes, there's a mathematical equation that says 2 to the 23rd, that's how many combinations there are. And what is 2 to the 23rd? It's about 8 million. And there's a number you need to know. There are about 8 million possible combinations of ways that your homologous chromosomes line up in metaphase 1. Meaning, for example, if we were to look at all your sperm and all your eggs, whichever you may be, if we were to look at 8 million of them, we statistically would only find one where it has where you would have all mom chromosomes and no dad chromosomes, or all dad chromosomes and no mom chromosomes, because there is a 1 in 8 million chance that would all line up that way. Another way of looking at this is if we were to look at your sperm and egg, it could somehow look at 8 million of them if we had the time to do that. For each one of those 8 million cells that we looked at, the way those chromosomes are lined up would be completely different. And I'll show you a picture. That'll make a little bit more sense here. 8 million different ways that you can produce sperm and egg. And considering that's just you, and then taking the account your partner also has 8 million ways to make sperm or egg, then when you combine that together, the possibilities of that particular sperm meeting with that particular egg, that means there are 64 trillion combinations of chromosomes. So if you and your sexual partner were to have sex 64 trillion times and somehow have a baby each time, or at least a cell we could look at, statistically, each one of those 64 trillion times it would be a different set of chromosomes. Never once the same. I mean, it's possible that it's the same, but statistically, that's how many different combinations there are. So when they say you are unique, you truly are unique. I always think about that too. I don't know if anyone else ever daydreams. You guys are a little bit young for that, but sometimes I daydream like, what if I did this different in my life? What if I did that different in my life? And then I think about how a small change would be a huge change. Like with my first daughter, if she was not fertilized at that very moment, right? If she was fertilized 10 minutes later, it wouldn't be her, right? Because there's 64 trillion possibilities of what it could be. So Anyway, that's big numbers. That just shows you how unique we are. And again, this is just talking about the way the chromosomes line up. This doesn't talk about anything else. We'll talk about other stuff here in a second. That's just the way the chromosomes line up. Let me show you a picture of this before I take any questions. Here we have a cell that has 2 pairs of chromosomes. And remember, we have 23 pairs of chromosomes. So imagine this is only 2. This is what I was getting at. I was trying to like mentally paint a picture for you. Now you can look at it yourself. Here, all the mom chromosomes lined up on one side and all the dad chromosomes lined up on the other. It just happened to happen that way. The only other possibility is here you have mom on one side with the dad on that same side, and then a dad on one side with the mom on the same side. You can see, right? Those are the only 2 possible combinations, right? It's pretty simple when you only have 2 chromosomes. So with those 2 possible combinations of the way they line up, we have these different combinations of sperm or egg. 1, 2, 3, 4, 5, 6, 7, 8. 8 different combinations. And that's just with 2 pairs of chromosomes. So imagine trying to draw that with 23 pairs of chromosomes. And that's why there are about 8 million ways you can produce sperm or egg, and 64 trillion ways that those sperm and egg can come together. It's really huge numbers. It's amazing when you think about it. Again, you are really unique. If you were not fertilized exactly when you were fertilized, you would not be you. Anyway, any questions about this? All right. So again, that's just the way the chromosomes lined up. 64 trillion different ways you and a sexual partner can produce an offspring. 64 trillion just by the way your chromosomes are meeting up in the middle in meiosis I. Then we go back to this idea of crossing over. So remember, crossing over is the exchange of corresponding segments between homologous chromosomes. So again, in your mind, just picture that picture that I showed you where the homologous chromosomes kind of playing footsie with each other and they swap apart. So then you have a mom chromosome with a dad chunk and a dad chromosome with a mom chunk. And remember, like I told you, it's completely random. Sometimes crossing over doesn't happen. Sometimes it happens more than once. Sometimes it's a big chunk. Sometimes it's a small chunk, all that. So not only do you have the 8 million different ways that you can produce sperm or egg by the way the chromosomes are lined up, not only that, but the ones that are lined up, they're not even completely mom chromosomes or completely dad chromosomes. They are some completely random version of a mom chromosome and a dad chromosome. So I'm going to go ahead and go to the next slide because this is information you've already had today. And I just want to talk about this with the picture up there. So again, there's crossing over. I like it. This picture is different than your last one because the last one only showed 1 chunk of chromosome being swapped. Here we have 2 chunks. So there we go. With this crossing over of these 2 chunks, we now have, let's just say blue is dad. I don't know. We have a completely dad chromosome. So this person, right, excuse me, here's the original cell up here. And it only has 1 pair of chromosomes. Keep in mind, we have 23 pairs of chromosomes. Anyway, this cell is about to go through meiosis to produce sperm and egg, or egg. So what happens here, we have a completely dad chromosome. Here we have a dad chromosome with a little bit of mom on it. Here we have a mom chromosome with a little bit of dad on it. And here we have a completely mom chromosome. And that's from crossing over. And again, those are the things that are going to randomly line up in meiosis I 8 million different ways. So again, I know I keep saying this, but the genetic uniqueness of you, it's quite amazing if you've never thought about it before. There is no other you. Anyway, unless you're an identical twin. But speaking of which, the identical twin part would have to happen after fertilization if it's like this, right? Because you would have to have the exact one of these sperm or egg combining with the other sperm or egg. And then that would have to split mitosis. But that's a whole different discussion too. You can look that up for independent work if you want to. The relationship between identical twins and mitosis as opposed to meiosis. So let's talk about when things go wrong. That's something we don't talk a lot about in this class, but if we had more time, we would. A lot of times things don't happen the way they should. Respiration doesn't always happen the way it should. Photosynthesis doesn't always happen the way it should. Meiosis certainly doesn't always happen the way it should, and that's what we're going to talk about here. Your next word for attendance is this. Errors. So anyway, what happens? What happens when something goes wrong in meiosis? As your book points out, it can result in genetic abnormalities that range from mild to severe to fatal. As a matter of fact, a lot of times, it kind of results in miscarriage. So a lot of miscarriages, a lot of people don't even know because it's such a bad abnormality. They were pregnant, and the thing miscarried before they even knew it was they were pregnant. But anyway, let's talk about these things. How accidents during meiosis can alter the chromosome number. Because that's the big thing here, is having the wrong chromosome number. How does it happen? It happens because of something called non-disjunction. That's one of the easiest words I think that we learned in this semester because it sounds like what it is, non-disjunction. So the chromosomes don't come apart the way they should. Either the homologous chromosomes in meiosis 1 or the sister chromatids in meiosis 2. For some reason, one set, they just don't separate. That is called non-disjunction. And obviously, if one of your 23 pairs of chromosomes didn't separate properly, that's going to give you some odd numbers. Some of your sperm or egg are going to have too many chromosomes. Some of them are not going to have enough, right? Because one of them, went somewhere else where it shouldn't have. As your book points out, it can happen in meiosis 1 or meiosis 2. Obviously, because like I said, it could be your homologous chromosomes or your sister chromatids that don't separate. If you're taking notes, I wouldn't worry too much about what's on this slide. Just know that what do we call it when they don't separate? We call it non-disjunction. That's the most important thing you need to know. And hopefully common sense can just tell you, and this is going to make a lot more sense right now when I show you this picture. Hopefully common sense just tells you, again, if these chromosomes are supposed to separate, but they don't, then that's going to give you some odd numbers. That's it. That's all you need to know. This is very specific and gives you all these different ways it can happen. I'm not going to get that specific. I just want you to know that if chromosomes aren't separating, obviously you're going to end up with some odd numbers. In this particular example, the non-disjunction happens between the homologous chromosomes in meiosis 1. What's the result? Well, this egg has one too many chromosomes. This egg has one too many chromosomes. This egg has not enough chromosomes, and this egg has not enough chromosomes. Of course, I said egg. It could be sperm, whatever. The gamete. Point is, if you have non-disjunction in the first round, it's definitely messing things up. There's a 100% chance if you have if you have non-disjunction in meiosis 1, every single one of your gametes is messed up. Now, if non-disjunction happens in meiosis 2, then we're talking about sister chromatids failing to separate. So meiosis 1 went off as normal, meaning you started with 2 normal cells, right? That one's normal, that one's normal. However, this one messes up. We have non-disjunction, so when it splits, then you have this gamete's messed up because it has too many too many chromosomes. And again, this one's messed up because it has too few, right? That's what non-disjunction does. But hey, here's the good news. Because it happened in meiosis 2, you do at least have 2 normal gametes. I probably won't ask this. Maybe it's a good extra credit question. But a good question might be something like, what would be more harmful? Non-disjunction of meiosis 1 or non-disjunction of meiosis 2? And the answer would be non-disjunction of meiosis 1, right? Because if it happens in meiosis 1, every single one of your gametes is messed up. If it happens in meiosis 2, half of them are okay. Any questions about this slide? All right. The next slide I'm going to show you, right? So what we just saw was the production of the gametes, right? We just saw what happens when something goes wrong in meiosis. The next slide is going to show you what happens when one of those gametes from that past slide fertilizes a normal zygote. So here we go. Here's an abnormal egg right here. It has an extra chromosome. Right here, that's the extra. And it's meeting with this normal sperm cell right here. What happens? You have an abnormal zygote that has an extra chromosome. And of course, there's the 2n+1 and the n+1 and the n. And don't worry about that. Don't worry about these details. It is just an example. The point here is if you mix an abnormal egg with a normal sperm, or even vice versa, you're going to have an abnormal zygote. Your zygote will have too many chromosomes or not enough. And in this particular example, it's too many chromosomes. What happens when that happens? You get something called Down syndrome some of the time. Down syndrome is something we call trisomy 21. That is when you have an extra number 21 chromosome. And what do I mean by number 21 chromosome? To remind you, we have 23 pairs of chromosomes, right? So it just so happens that the 21st pair, if you have an extra one, it's called trisomy 21. Because instead of having 2 of them, you have 3, hence trisomy 21. So again, instead of having 46 chromosomes total, you now have 47 chromosomes. And when that happens, that is what we call Down syndrome. So now you know what Down syndrome is. The last word for attendance is going to be mother. So when we get back, we'll talk a little bit more about Down syndrome. Another good independent work topic is this topic right here. I'm only giving you, I think, 1 or 2 examples of chromosome abnormalities, but there's a lot of them. So you can look that up. What happens if you have extra of these chromosomes or too many or too few of these chromosomes? There's so many. I learned about a new one just the other day. I have a friend who has a kid that has autism, and they just did a genetic testing, and it had to do with chromosome numbers. Anyway, that's it for today. Any questions? I hope to see you in lab. It's an easy lab, but it's important that you're there and you're there on time. If you're late, you will lose points. If you leave early, you will lose points. If you don't show up at all, you're really going to lose points.
Thank you. See you there.
All right. You guys have a good day. Don't forget to send your emails for attendance before 9 a.m.